In the last video, we started our look at viruses, and we're going to continue that in this video, taking a look at the viral life cycle. So we know that viruses must infect a host cell in order for them to metabolize and reproduce. Viruses in their genome do not contain all of the instructions and all of the enzymes that are necessary for them to complete their life cycle. So when they infect a host cell, they basically take over the host cell's machinery in order to produce more virions or viral particles. So what we're going to look at in this video is how the virus is actually able to accomplish that. So our first thing that we're going to look at here are life cycles of viruses with our prokaryote host. So we're going to take a closer look at the life cycle of bacteriophages. Remember, these are viruses that can only infect bacterial cells. Since these bacteriophages only infect bacteria, they can provide a good model for us. We can study how bacteriophages infect these bacterial cells and what they do and how they do it, and that provides us a good model for understanding viral life cycles in other cells. Now, when we look at bacteriophages, we have a couple of different types, virulent phages and temperate phages. Virulent phages typically are going to cause the cell to die because they're going to cause lysis, or they're going to cause a rupture of the cell so that all of those new viral particles can escape. Temperate phages do not typically do that. They can become part of the host's chromosome, and every time the host replicates, it includes a copy of that viral genome as well. So in the lytic cycle of the virulent phage, so we're going to look at the virulent phage that causes lysis. So during the lytic cycle, the bacteriophage is going to take over the cell, cranks out new bacteriophages, and then destroys the cell as all of those new viral particles erupt. So here are the 5 stages in this lytic cycle of the bacteriophage. So the first thing that happens is that the bacteriophage has to attach to the surface of the host cell. So it's going to use those little tail fibers for attachment. The second step is referred to as penetration. In this stage, the viral genetic material, DNA, is going to enter the host cell. Notice that the bacteriophage itself, the virus itself, does not actually enter into the host cell. Only the genetic material enters the cell. Then we have biosynthesis. This is when the phage starts to replicate, the DNA starts to replicate, and all of the different components of the phage proteins are made. And then maturation is when they are assembled. So now the capsid and the DNA and all of these other pieces are now assembled into the complete bacteriophage, and then we get lysis. All of those new bacteriophages now explode out of the cell, and they're going to go on and find new host cells and start this cycle over again. Okay, so this is the lytic cycle because it causes lysis of the host cell. Now, in the lysogenic cycle, okay, we also have attachment and penetration. So we have attachment of the bacteriophage to the host cell. We have the penetration, the DNA that has been injected essentially into the host cell. But what happens is that the phage DNA actually becomes incorporated into the host genome. This is called the prophage. So this is the actual DNA of the virus that is now becoming incorporated in the DNA of the bacterial cell. As the cell divides, the prophage DNA is going to be passed on to all of the daughter cells. So from this point on, every time this bacterial cell divides, the daughter cells are going to contain this prophage. They're going to have this extra piece of genetic material that belongs to the original virus. Now, this can continue on for many generations. Okay, and it this DNA essentially just remains latent or inactive, um, and then at some point, the prophage DNA is going to be excised. It's going to be taken out of the bacterial chromosome and then enters into the lytic cycle. So the phage DNA starts to replicate, make the components are assembled, and then we eventually get to lysis. But this first part here, this replication part, can go on for many, many, many different generations. Now, this insertion of the prophage can alter the bacterium. It's bringing in extra genes, and some of those genes may increase bacterial virulence. This is called lysogenic conversion. That this bacterial cell is now different and has different capabilities, different components than it did before it was infected with its bacteriophage. For instance, Clostridium botulinum, less virulent in the absence of the prophage. So if it has not been infected with the bacteriophage and had that DNA inserted into it, then it is less virulent. It's not as able to cause disease. So in some cases, the infection with this bacteriophage allows the bacterium to become more virulent, more able to cause disease. Occasionally, during the viral life cycle here, we can have something that occurs called transduction. Transduction is when the bacteriophage, the virus, is going to transfer bacterial DNA from one bacterium to another during sequential infections. So we have two types of this, generalized and specialized. Generalized is when a random piece of bacterial DNA is transferred by the phage during the lytic cycle, and specialized transduction occurs during the end of the lysogenic cycle as the phage is entering into the lytic cycle. Essentially what happens is this allows for some genetic recombination, that now our bacteria have bacterial DNA. They have portions of a bacterial genome, and it can give the bacterium new characteristics. It's something that it hasn't previously had, and it's almost like sometimes this transduction occurs by accident. A mistake is made, and in the capsid we get a little piece of the bacterial DNA. But sometimes, especially if we're looking at the lysogenic cycle, when the prophage is excised, a little piece of the host cell DNA is excised with it. So it basically allows some genetic recombination that would not be able to happen otherwise. Bacteria do not reproduce sexually. They are they strictly function in asexual reproduction. So they're essentially clones of one another. And the only way that they can get new DNA is to be infected with a bacteriophage. Now, if we switch gears a little bit and look at the life cycle of viruses with animal hosts, we see a little bit of a difference here. Now, lytic animal viruses have a life cycle that's very similar to the lytic cycle of bacteriophages. Attachment, penetration, biosynthesis, maturation, and release. It's fairly similar. However, the mechanisms for penetration, biosynthesis, and release are going to be different when we're comparing our bacteriophages and our animal viruses. So if we take a look at this figure here, we've got an animal cell, we have the influenza virus. And so the influenza virus is then, it attaches, so we still have to have that attachment process. But in this case, the cell engulfs the entire virus and brings it into the cell. So we're not just injecting the viral genetic material, the entire virus is incorporated into the cell. Then we get this uncoating here where the viral RNA is going to be released. So the genetic material for the virus is released. And then that viral RNA, in this case, enters into the nucleus. It's replicated by a particular enzyme called RNA polymerase that's used to make the viral proteins. We get the assembly here of the virus, and we basically get a situation where only one virus at a time is leaked out of the cell. So this host cell is not killed. It's going to continue to be a little virus factory. It's going to continue cranking out new viruses until the cell just gets worn out and it dies on its own, or the immune system flags it for destruction and it can be killed, or we take some type of antiviral medication that is going to kill these virus infected cells. So the cell just becomes a little virus factory until it dies in one way or another. Something else that we have to consider is what type of genetic material is found within our virus. This is going to affect exactly how the assembly process, how that biosynthesis and assembly process works, because depending on the type of genetic material, whether it's DNA or RNA, or whether it's single strand or double strand, we may have to make some conversions to a different type of genetic material before the viral proteins can start to be assembled. So this is something else that we see that's different between the bacteriophages and the animal viruses. Now, some viruses have the ability to cause persistent infections. This is when the virus is not completely cleared from the organism and it can stay in certain tissues or certain cells and just may remain silent, or it may actually start to replicate and undergo infection without harming or killing the host. We can have latent infections and chronic infections. In latent infections caused by these latent viruses, they are capable of remaining hidden inside the cell. They can typically cause an acute infection, so an individual will have an acute infection, and then the infection becomes dormant, and you think you're over it, you think it's cleared, but it's just going to remain latent. It's just going to remain hidden. And one of the best examples that we can see of this is with the varicella zoster virus. Individuals contract chickenpox when they're a kid. That's the acute phase of the infection, and then it goes away. Well, that virus hides in nerve cells, and years later it can become active again, resulting in shingles. A chronic infection is when the symptoms are recurrent or persistent over a long time. If the body can't eliminate the virus, then the infections can become chronic. HIV is an example of this. It can become latent and then develop into this chronic infection. The untreated patients may experience no symptoms at all for many years, and then start to see those classic symptoms that are associated with HIV and AIDS.